And without much further ado, I would let Julie take the virtual stage and do the talking.
Thanks, Harlan. So if you have questions during the talk, you can type them in and Harlan will read them to me. We don't have to wait until the end of the webinar to answer the question. I think it's more interesting for everybody if this is something more interactive. Yeah, I have a hard time with the concept of webinar where I just talk to my slide and I don't see anybody. So please participate in this webinar to make it more interesting. So as Harlan said, my name is Julie. So I joined Singleron 3 years ago. Yeah, so I joined the Cologne site in Germany first, but recently I moved to the US. So we are currently located in Ann Arbor, Michigan. So without further introduction, so here is Singleron. So who we are. So we are a global company. So we have locations a little bit everywhere in the world. We have 2 big locations in China where they do production, service, development of new technology, both at the molecular biology level and at the engineering level. So I will talk a little bit about our instruments, so how our engineers are automated the technology and making our life quite simpler by doing so. We have a site in Singapore. They're mostly a CRO, so they process a lot of sample for clinical application. But they will do any kind of different service, and they have like also kind of act as a distributor for this region. I already mentioned that I joined Singleron Cologne, Germany. So if you have a chance to visit, it's a beautiful city. So what did they do in Germany? So again, we do product development, service, and as Arnin say, a lot of the cell people are located there for Europe. I recently moved in December 2023 back to Michigan. So Michigan is kind of home for me. So it's where I've been living for more my adult life. So what do we do in Michigan? So we do a lot of manufacturing. So a lot of our suppliers are American. So I will purchase different material from the supplier and we will package that in kit for you guys. Or we would use the reaction or the material to do service. So if you don't want to do single cell yourself, you can always ship your samples to us, and then we can proceed accordingly and give you the data. And nonetheless, we are also a site in Connecticut where they develop new technology. They also have an awesome FAS over there that can support you in any of your questions. If it's your first time doing single cell sequencing and you're not quite sure how to proceed, they're awesome consultant and you can talk with them. They can also show you how to work with the technology itself. Like if you buy the reagents, and then they can basically give you an introduction of how it works, go over the protocol together, and make sure that you are successful. I always start this talk with this figure. And until recently, I had no idea where I took this figure because it was such a long time ago. And then this morning, I remember that Google had this feature that you can search by images. So then I just put this image in Google, and then I found out that this is an image from a paper from 2017. And why did I put that there? Because it's so outdated. This was basically the paper that incited me to go in this field. So I was reading the paper, and at the time I was working on non-invasive prenatal testing, making new tests for this kind of application. And then I read this paper, and I was like, "This is awesome! Like, we can do this kind of technology. We can. There's so many applications to that." And I was particularly interested in the fat because for me, fat was just like an energy reservoir, and that was just like adipocyte. But no, there's so many different cell type, as you can see here in the figure. Yeah, so anyway, so this 2017 paper, which is quite outdated though at this time, was the reason why I moved to this field. It took me a bit. I only moved to this field in 2020, but nah, I have no regrets for sure. Okay, but what I want to say here is like, what is the main application, or why do we do single cell? Why do we study cell at a single cell level? And single cell sequencing is now a commodity. I'm not going to lie to you. It's not a new technology anymore, but the applications are growing and growing because we can talk and, it's basically coming from you researchers. And you can find new application to this kind of technology every day. But anyway, for the people who are just joining and are new for single cell sequencing, the main goal is to identify the different cell type in a tissue. So it's a given, like the example of the fat. So there's more than adipocyte in the fat, and then we know about them. It's also to understand the differential expression level in response to treatment or disease, and then we can compare inside a tissue the expression change in a cell cluster. So if you're interested in B cell, for example, then you can see the differential expression only in B cell, not like in fibroblast or endothelial cell, but you can really focus on the cell of interest because we can tell them apart. We understand better the difference between patient heterogeneity and to drug response. And this is a big application to cancer or inflammation or autoimmune disease. And I can get more into that later. And we can study how the cell responds to each other or to their microenvironment. And it's not only about tumor microenvironment, which is a big splash those days, but it's also about pathogen, for example. So we had a customer that was interested in salmonella, and she was studying some virulence factor versus what's happening inside of the cell. So she could basically link together those bacterial genes with basically what was happening in the gut of humans. So super, super interesting application. And again, those applications are not coming from us. We make the technology available for researchers, but how you apply it is definitely up to you. Single cell has been a key factor in cancer research. So we can study and understand better the heterogeneity of cancer cell. So inside the same patient, you will have different cells that can go into metastasis and invade different tissue. And in another patient, even if it's the same cancer type, it won't happen. And understanding why those things are like that is very important to understand how we can fight cancer. Tumor immune microenvironment has been also a huge application in single cell. So it's very important to know what are the cancer cell doing, but also what is the immune system reacting to them. Is the immune system active? Like how many T cell, B cell are in this mixture? Are they active? Are they dormant? What can we do about it? And I think one of the last application is the immune checkpoint inhibitors. So those, not that new anymore, but those drugs have been helping so many different patients. And what do they do is that PD-1 or PD-L1 inhibitors, what do they do is like they basically keep our T cell active so they can slow down or kill this cancer cell. And those drugs have been saving so many lives. So anyway, so single cell sequencing in cancer research, it has been the main topic, but it's not the only topic. To finish this introduction to single cell, I would like to briefly talk about the UMAP, because every paper that we'll publish on single cell will have this UMAP. And I don't know like the background of everybody in the audience, but I just want to very shortly introduce what's a UMAP. So in short, very short story, a UMAP is just a way to plot the data in 2 dimensions. Because the single cell data, ideally, we will look at it in 3 dimensions, but nobody can publish that. So basically a UMAP is just a way to reduce 1 dimension. Here, on the right, you can see an example of a UMAP from PBMC. So you have a lot of different B cell, for example, and T cell, and all the different dots will represent a cell, and all the different color will represent a cluster. And then you can identify how many, for example, B cell you have in this cell population, how many T cell you have, and if you had 2 samples, you could compare them. You can compare the B cell from sample 1 to the B cell of sample 2. So this is very interesting. But the question remains is like, how do you identify those clusters? So we know we do sequencing and we see like gene expression, and the cluster are basically grouped together, like the cells are grouped together if they are like each other. But once you know that, how do you say, oh, this is a B cell, this is a T cell? So we have this matrix, and here it's very simplified. But basically we have in the matrix, like the genes that are expressed in those different clusters, and they can be on or they can be off. And this is this representation here of the Violin plot. So what is a Violin plot? So for example, if you look at the third column of the first row, you can see this is the expression of CD79A in this cluster. And the Violin plot will represent basically all the cells in this cluster. So if we have 1,000 cells, then this is basically the average of the expression. And we can see that very little of those cells don't express CD79A because the bulk, or where the Violin is the largest, is higher. So the first cluster, basically none of the cells are expressing that gene. And with the literature and with different database and with different tools, we know that those two genes, for example, are markers of B cell. So then you can tell that the third cluster will correspond to B cells. And it would be the same story for the natural killer. So we know that GNLY and NKJ7, they are marker of natural killer cells. And you can say that the two last clusters are expressing those two genes in abundance. So most of the cells in those clusters are having a high expression of those two genes. And then you can identify those cells. So this is a manual annotation. Like most people don't do that anymore because the database are getting better and better. And there's more tools to do that. But this is basically just to explain the concept. And there's something to say about manual annotation, because as a researcher, you know your system better. You can go into the literature and do like sub-clustering, for example, and go really into the details. So this is still a useful tool. Okay, so I want to jump into the Singleron technology. Yeah.
I'm sorry to interrupt. I
No, go ahead.
see a mess. I just wanted to actually bring into notice a point that Sarah brought up, that can you hide the notification at the bottom of the screen if it's
Yes.
possible? Yes, great,
Yes.
perfect.
Sorry, I
Thanks.
didn't even notice it was there.
No worries. Yes.
Okay, so this is a single cell workflow for RNA-seq or other application, to be honest. So we most of the time we're starting from a tissue, and the goal is to generate this single cell suspension where every cell is basically individual, like they don't touch each other, they're not linked to each other. And then with this, we can load our microfluidic chips and do the cell partitioning, capture the messenger RNA, and the rest is basically common to every other platform. So we do a reverse transcriptase, we amplify the cDNA, and then we sequence. We have two instruments that can automate those processes. So we have the Python and the Python Junior. So it provides a tool to dissociate the tissue into a single cell suspension. So you basically put your tissue in this little flask here, and then we add enzymes, like an enzyme mix. And then the enzyme will basically soft the tissue and digest some of the link between the cells. And then the mechanical action inside the instrument and the eating action, because all those enzymes prefer to work at 37 degrees, will help with the dissociation. So it's a mixture of enzymatic dissociation, as well as mechanical dissociation. So I don't know if you can see in the picture, but there's little palms in the unit, and it acts a little bit like a pepper grinder. Okay, so once you have your single cell suspension, we do the cell partitioning. And again, we have another instrument that you can use for that. It's called a Matrix Neo, and it's one of my favorite instruments. It works fairly well. And then you can automate this whole process, which I like. It's just faster, and you can do 4 chip at the time, so 4 samples at a time. The chip, you could load it manually. It has a little port here that you can use a P200 pipette to just put your cells in, and it works really nicely. I will show you in a couple of slides. So how it works. So this is a chip. The chip is really the core of the technology at Singleron, and it's what is differentiating us from other technologies. So how does it work? So you basically inject your cells in the chip, and the chip has 150,000 wells. So that's our standard chip. We have different chips. Like we have a bigger chip that has 250,000 wells. In R&D, we have even like one that had 500,000 wells. So it becomes a little bit overkill at this point. But anyway, so it's very flexible. And it has like, and of course, even if the chip has 150,000 well, that's not what we want to capture. We don't want to capture 150,000 cell. It would cost so much in sequencing. So we only populate about 5 to 10% of the chip. So we capture basically between 5,000 to 10,000 cells. The cells are injected and they fall down by gravity in the chip. And then everything is washed away. Like if we have floating debris, for example, they wash away. And then we will add the beads. So the beads are magnetic, and then they will populate the whole chip. And once you lyse the cell, because the beads have this nice oligonucleotide, they have multiple, this is just a representation, and the oligonucleotide has a poly T at the end. So all the mRNA that have a polyadenylated tail can be captured by the oligo on the beads. And because every bead is a little bit different, because it has a barcode, so every bead will have multiple oligo, and all those oligo will have the same cell barcode. And then we can recognize, for example, this orange cells here will have all the RNA or all the mRNA from this cell will have the same barcode. And then we can say, oh, it came from the same cell. I'm not sure I just mentioned that, but we have 1.7 million different barcodes, and the chip is 150,000 wells. So that the chance that 2 barcodes will capture a cell is almost impossible. Okay, so I want to share a little bit of the experience that I have with the service. So when I introduced myself, what I did not tell you is that when I started at Singleron, I was working in the R&D department, so developing new product. However, I transitioned to service. First, I was not super happy with this transition. I was thinking that service would be a little bit boring, but in fact, it's quite challenging and it's quite interesting. So in R&D, we work a lot. We're like super focused on a couple different projects, and like we can go very deep into all the detail of those things. But with service, we have so many different projects, so many different customers, so many different applications. So you cannot go deep in anything. But there's so many different project and application and challenges related to all those different needs, and it's quite interesting. And I was quite happy after the first year of transition, I have to say. And it gives me the chance to talk with customer and learn about their project, which I always think it's interesting. But anyway, so my experience with the service is that the input is key for single cell. A good input will lead to really good data. So what do I mean by that? So when we receive a sample, for example, that has a lot of necrosis, so we work a lot with a lung sample from human, and if they were smoker, there's so much necrosis. Like basically we had samples that were all black, and there's nothing we can do with that. So a good and healthy sample is key. The presence of blood, so we have to wash as much blood as possible, because even if the erythrocytes don't have a nuclei, there's still a lot of the globin gene in there, like in the mRNA level, and it can pollute your signal. So you basically eat your sequencing reads. So you want to remove as much red blood cell as possible. And the quantity and the quality, of course. So there's some tissue that has a bigger, like a better density in cell than others, or the cells are just easier to get out. So then you don't need that much. Like, for example, we can work with 50 milligram of lung or kidney. However, when you start working with skin or fat, for example, then you need a little bit more tissue to achieve a really good single cell suspension before going into the workflow. So it's all details that are super important when you talk to the customer or when you prepare your experiment. So to alleviate some of the logistic problem, we came out with a tissue preservation solution so people can ship or pause their sample after collection. So for example, if you have a sample and you want to just wait another day to obtain another sample so you can do all the experiment together. So this is possible. Or if you want to ship the sample to a core facility or something like that, you can immerse the tissue in the tissue preservation solution and it will keep its integrity, at least for like 48 hours. And this is what this data is showing here. So you can see that we compare fresh tissue from the mouse skin that was never in the tissue preservation with some of the tissue that was immersed for 24, 48, or 72 hours. So up to 3 days. And here we look at the gene expression in the different clusters. So for example, the fibroblasts here, that is the first cluster. We look at the top 2,000 genes that are differentially expressed with the other clusters. And you can see that between the fresh sample and the 24 hours, 48 hours, or 72 hours, the Pearson correlation is really good. So the gene expression is conserved. So closer this number is from 1, better it is. However, you can see that other cell types are starting to get affected, like for example, the sebocytes. They are affected after 3 days in the tissue preservation. So I would suggest like 48 hours is a good time. So a good threshold. And you know, those days it's pretty easy to ship a sample and it takes less than 24 hours. So then if we receive a sample, we just proceed right away with the tissue dissociation. So usually there's no problem unless there's a snowstorm in Michigan. Okay, so tissue dissociation, we also have a solution for that. I already talked to you about the Python and the Python Junior. So we have this pepper grinder that we can use. But what do we look like? What does a good single cell suspension looks like before you go into the rest of the workflow? So you have to look for cell aggregation. So if you have clump, it's not good. It may clog the workflow. It may cause like doublets. So you have to make sure that the cells are not tied to each other. You have to sometimes do a debris removal, like it's the case for removing the myelin from the brain, for example. Otherwise, it kind of create a mess in the chip. We talked already about the red blood cell. We can lyse them. There's product out there. It's pretty simple. You can get rid of them if they are in your single cell suspension. You don't want to sequence dead cell. And to have a good dissociation helps with that. So you have to be gentle. You don't want to break the integrity of this membrane. So you have to be gentle when you do the dissociation. So there's also kit now that you can use to remove the dead cell, but it's better to just upfront try to be gentle, and then you don't introduce bias with those different steps. And accurate cell counting, that's key. I talk a little bit about Poisson distribution. So what happens if you load too many cells? Well, you will have doublets. You will have data that is diluted. And if you don't load enough, then you won't have enough data to have a successful experiment. And you may miss some of the cell cluster because there's not enough cell to form a cluster. So it's very important to load the right amount of cells on the chip or any technology. Here it's just a graph that shows a comparison of the cell viability between different commercially available dissociation mix. And you can see that the JetScope, which is a Singleron tissue dissociation mix that is available commercially, we usually have a good viability of the cell. We also have a new product that aim at conserving the epitope at the cell surface. So it's a very, very gentle dissociation, and it's aimed at people who do fact sorting. So then they can have a better experiment here, like the sorting can happen better. And a lot of people really do fact sorting before single cell. So it helps removing some of the debris. It helps concentrating on the cell of interest. So this is also an application. This is paired together with single cell sequencing. Here I want to go into more detail about the chip. So this may be a little bit new for you. I know that the leader in the field is using droplets. So using chip is particular to Singleron. So this is how the inside of the chip look under the microscope. And it's very nice because when you do manual loading, when you use your P200 to load the chip by this little port, you can look at it under the microscope. It's not a black box. You can know exactly what's going on in the chip. So for example, we try to load 10% of the well with cells, and you can see here the little cells in the well. And then we add the beads. And there's only one beads that will fit in one well because of a size restriction. So the wells are about 40 micron and the beads are about 30. So you cannot have two beads in one well. The beads, as I mentioned before, they are covered with oligonucleotides, which the 3' end as a poly DT, and then we can capture the messenger RNA that way. This is a picture of it. So it is basically a big focus on the oligonucleotide on the magnetic beads. So we have a PCR indel, so we can use that later on to amplify the product, the cDNA. We have a cell barcode, so every bead will share the same barcode, and we have 1.7 million different one. We have UMI, so unique molecular index. So those are degenerated, so they're different on the bead. So then we can recognize later on if the capture molecule, once it's amplified, is it a different molecule or is just a PCR duplicate? So it helps with the accuracy of counting your genes.
Really? I'm sorry to interfere again, but we do have a question right now.
Okay.
Yes, and I would like you to answer it. So the question is there a way to load the sample on the chip for the second time if you notice that the cell coverage of the wells is low? And do we recommend this or we don't recommend this for cases where the number of cells that we have captured is not up to the mark, for example?
Yeah, of course. So because it's such an open system and you can do basically whatever you want, so what happened here is that you will load the cells and they will travel in the chip. And here we have an outlet. And then everything that you load in the chip will eventually come out of the chip because you keep adding things. So there's nothing preventing you to take the output and put it back. And you can do that as many times as you want until you're satisfied with the loading. Here it's more the molecular biology behind the technology. So once you are in the chip and you capture your messenger RNA or everything that is polyadenylated, what we do is a reverse transcription. So the poly T will be extended and it will be a duplex RNA DNA that is formed. That DNA is now covalently attached to the beads. And then we do PCR amplification. This technology here is based on template switch. So this, I don't know if you're familiar with template switch, but it's not like we don't own this technology. It was published in 2001, I believe. So it's quite old now. But basically, we use a MMLV reverse transcriptase for the extension, and it will add some G at the end. And because the template switch oligo has a couple of C, then they will anneal and extend again. So, and then we can use that for a PCR after, because we have a PCR indel on the beads and we have this TSO added at the end of the molecule. Then we can amplify the cDNA that way. So it's been used for years. And then this molecule now have the cell barcode. And then we can just make library prep out of it. The kit is containing library prep for Illumina, but we have customers that have been sequencing those library on Element or other sequencer. You don't have to fragment the cDNA if you want to do long reads. You can use PacBio, for example. But for Illumina, since it's a short read sequencer, this technology here that is represented has this fragmentation of the cDNA. This is what's happening on the sequencer. If you use Illumina or Element, for example, it's a paired-end technology. So the first read would be used to sequence the barcode and the UMI, and the second read would be used to basically identify your gene of interest. Yeah, it's pretty simple, and it's pretty, I mean, most of those technologies are using the same principle. There's nothing new there. We have software that enables you to analyze the data. So the first part of the software is basically taking the raw files from the sequencer. So it can be BCL, it can be FASTQ files, and then it will align those reads to the genome of interest. So if you're using mouse, then you will use the mouse genome, of course. And then it will create those matrix, the matrix that we already talked about. So this one is a little bit different. So it will have the gene in the first column and in every row, basically. And like the cell with the level of expression will be like in every column. So it's basically a giant Excel sheet. And then with this expression matrix, you can cluster the cells that express the same gene. So they will be really tight together and they will cluster together. And then you can do annotation. So you can identify your B cell, your T cell. So you can do that manually. You can use different tools. We also offer this database that is curated. It's called Synecosis. And I believe right now we have free, Arlen, you can correct me, but we have like a month free if you want to test that. Celescope is completely free. It's available on GitHub. Synecosis is a database that is for some costs. Anyway, if you have any question about costs, Harlan, which is our cell person, can answer those questions. Matrix Neo is the way we automate the chip loading. Again, you can do 4 different samples at a time, and it's really fast and it's pretty convenient. So for service, for example, we use that all the time. And here's just to show you the reproducibility of Neo. So we loaded the same, basically, cell suspension 4 times on 4 different chips. And basically, it looks exactly the same. So this is the cell representation here. So this, of course, there's a little bit of difference because the cell that you load from the cell suspension may be a little bit different because you take an aliquot of it. But it's really well conserved. Another question you may have is say, oh, you have manual loading and you have matrix loading. Are they different? Short answer, no, not really. We have some data that we, again, took the same cell suspension. We loaded on matrix or we loaded on the like just manually, and we can see the same representation of the cell. So here's an example of a mouse kidney, and you can see like in blue it's the matrix, and in pink it's the manual loading, and the cell representation for every cluster is pretty much the same. We did the same for the spleen, and again, it's pretty tight between the two. This is triplicates of everything. I have to brag about my team because we offer service. So I still lead the team in Germany for the service. So we have a lot of different applications, a lot of different projects that we've been working on, and yeah, different cell types, so species. So if you have any question about how to proceed with some special sample, I mean, everybody can do PBMC, but it becomes a little bit more complicated when you have a project. Like we have different species that we work with, like Drosophila or C. elegans and plants. And so, you know, everything needs a little bit of finding. So if you have any question, please don't hesitate to contact us. We can help. This is an example of a tricky sample that we had a couple of years back already. It was a nasal biopsy. They were studying like basically some of the out COVID patient or the patient didn't have COVID at the time. They had COVID like a couple of weeks before. And they were trying to determine like why they lost the sense of smell. And then they were looking at the receptor of the nasal biopsy. So then, so that was super interesting project that we did, and this is published, so I can talk about it. But yeah, some of the sample were quite tricky. So you can see this is the biopsy, and we cannot even measure how many milligram of tissue we had to work with. So that was quite challenging, but I think the data looks quite well. So at least the customer was happy. Another question that we get a lot is that should I do single cell or should I do single nuclei? And I think it's something you have to think about logistically. Single nuclei is really cool because you can freeze your sample and you can postpone your experiment when you're ready. You collected all the tissue you wanted, and then you can have a better collection. For single cell, you have to do it right away. Like you cannot just preserve those things. So that's logistically, single nuclei, it's easier. But there's less RNA in the nuclei than in the cell. So then you may have a little bit of loss of sensitivity for your data. There's also consideration to take in with the size of the cell or how they behave during dissociation. So that can create some bias. So for example, for the liver, hepatocytes are very difficult to dissociate. And sometimes they can be quite big and they don't necessarily fit in the well of the chip. So then single nuclei would be more appropriate to study hepatocytes in the liver. But then if you're interested to study the immune system in the liver, then single cell is definitely the way to go. Same story for the mouse brain. So if you want to study neuron, because they have extension like axon and dendrites, so you better go with nuclei. So this is the representation here of the neurons. And in single cell, you can see we can barely capture any. So again, consider it. I mean, if you have enough money, you can do both because they're quite complementary to each other. But yeah, again, it doubles the cost for everything. Just maybe I don't have much time to talk about that, but we have an option for large cell. So here we study a model for human skin. And once it's a collaboration with a team in company in France. So they gave us some model skin. It's called EpiSkin. So people study skin. They look at the toxic effect of some of the product, like in solar preventive thing, or anyway. So instead of using rabbits or animal testing, they do this kind of model. So when we dissociated the tissue or this model, what we observed was really big cells. So this scale here is 50 micron. So you can see like some of the cells are almost like something like, they're fairly big. They're almost like 50 something. So then when we loaded the standard chip, the cell fit in the chip because it's 40 microns. So there's no problem there. But then when we tried to put the beads on top, then it did not fit. Like you can see all those wells that are occupied with cells, they don't have beads. So that doesn't work. We don't want to do bulk sequencing. We want to do single cell sequencing. So we have a chip that has a bigger diameter of wells. So this one is 40 micron. And then we can have a chip that has 100 micron. And then those cells are going to fit easily in the chip. And then the beads, instead of having 30 or 35 micrometer, they have 60. So then again, here we can see a couple of them are broken, but most of the time, only one bead will fit in one well, as the same principle. We have also multi-omic applications. So yeah.
Sorry, Julie, to interrupt you. We have one question from Ferdinando asking about Neo. So I thought maybe we can address this before we move to the next section. Yeah. So he has asked us about when you run 4 triplicates on Neo, do you need to integrate data with specialized bioinformatic tool to make a well-harmonized UMAP plot, or do we see no batch effect?
No. Sorry. Yes and no. So Okay, I won't go back to the slide, but yes, you can always apply batch effect. In this particular data, we did not apply it because we really wanted to know the robustness. So this is basically just data, like if you had analyzed those individually and put them together after that. So that we did not use batch effect. Should you use batch effect? Yes. Multi-omics. Yeah, so the technology enables to work with different multi-omics. So I think a couple of years ago, I think in 2018, the CiteSeq paper came out when you can study at the single cell level um, uh, protein at the cell surface, as well as the transcriptome. So that was one of the first application of multi-omics, and our technology is compatible with that. But in addition of that, we can do different things, which is a little bit more particular to Singleron. So we can mix and match some of the target. If you are interested in somatic variant, for example, we can target some of the gene not only by their 5 prime, but also, 3 prime, but also in the middle of the gene. So you can sequence basically an exon that you would be interested in for splicing variant, for somatic variant, for anything you would be interested to know about something that is not only at the 3 prime. We can also study glycosylation at the cell surface. We can look at the immunoprofiling, and we can also do some temporal resolution. What does that mean is like we can differentiate the RNA that is newly synthesized before and after a treatment. So this is all different application that we can do. I just wanted to start with chromoscope, which is the assessment of the glycosylation at the cell surface. The application for that, it's mostly looking at cell-cell interaction, but also in the context of oncology and autoimmunity. So all the immune systems are going to be affected. And it's known that the glycosylation at the cell surface is involved in those processes of recognitions or unrecognitions. So how does it work? So we have basically glycosylation at the cell surface, and we have this enzyme that is a modified enzyme, of course. It's a fucose. And the fucose has affinity. So it's a fucose enzyme. And the fucose enzyme has affinity for fucose at the cell surface. So then it will find its target, and then we use click chemistry to make a covalent bond between the enzyme and the modification here at the cell surface. The enzyme is also linked to an oligonucleotide that has a barcode and a poly A. So later on, it will act basically as a messenger RNA, or the same way if you want. And then we can quantify it. So then we incubate the cell together with what we call the chromoscope tag, and then link them, wash the excess so it becomes quantitative. And we do the partitioning the same way on the chip. And basically, yes, so this is the bead that is capturing both the chromoscope tag to quantify the amount of fucose at the cell surface, as well as the messenger RNA. And then we can analyze the data. And this is an example. So this is the level of glycosylation for different cell cluster. And one of the applications is to study granulocytes. So granulocytes here are depicted by the GMP. And you can see that we have different clusters. We have this cluster, and this little cluster there. And if you look at the glycosylation of those GMP, they're quite different. Like here you have more glycosylation, here you have spotty glycosylation. And then when you look at the literature, glycosylation of the granulocyte is basically an indicator of their maturation. So you can see here the little fucose that is represented by the triangle, the red triangle, basically disappear at the end. So we can think that those granulocytes are more mature than those here, for example. And it can all be proved also with the messenger RNA, because the messenger RNA are going to be different depending on the maturation, of course. But it's something that goes basically on top of each other, and they are really complementary information. We also can do a targeted approach to study fusion, for example, splicing or somatic variant. So we have different panels. So because we can design the oligo on the beads as we wish, we can put probe also on the bead, not only the poly-DT. So the poly-DT will capture the mRNA, but those probe will also capture some of the mRNA by the middle of the gene. And then we can look into different transcript by the middle or a little bit closer to the 5'. And this is an example here of a data. So we have a mixture of 3 different cell lines, and some of the cell lines are having somatic variants. So here it's an oncogene, so KRAS, and you can see that KRAS mutation are only present in NB4 and CCRF, and we can only see A18G mutation in NB4 and the G12 mutation in CCRF. So the sensitivity and the specificity is quite good. You can do the same with fusion genes, like, here's a tumor suppressor, so you can think TP53 is way less expressed in those cells than KRAS, which is highly expressed. But you can see the same data, basically. You have a mutation detected, and it's very sensitive and specific. And here you can do the same for fusion. So BCR-ABL is like the Philadelphia chromosome. It's involved in leukemia. And you can see that it's only detected in this cell line K562, not on the other. And it's the detection is really specific and sensitive. We did a dilution. So we diluted 2 cell lines together at different ratios. So 10 to 1, 100 to 1, 1,000 to 1. And if you capture around 6,000 cells, you can see that those ratios are conserved, and only the variants are found, or this translocation is found only, for example, in 10 cells out of almost 6,000. So it basically preserves the ratio. And so this is another application. We also have an immuno application, so we can look at the CDR3 receptors on T cell and B cell. There's a ton of application for that those days, including the CAR T therapy. But basically, the way we have 2 different products for that. One is basically another one of those targeted approach where we put a specific probe on the beads. In this case, we will target a constant region of CDR3. To amplify or to enrich for the receptor. And in the other case, we go full length, which is a quite more eloquent approach, where we capture the receptor still by the poly A of the messenger, but then we will amplify it by the constant region. And to do so, we have to invert basically the orientation of the molecule, because the constant region is very close to the 3' tail, the poly A. And then to be able to basically put the barcode closer to the variable region, we make this circle, and then we reopen it. And when we reopen it, the orientation is changed. And then the barcode is closer to the variable region. So it seems quite complicated, but it's very simple, in fact. And the data looks pretty good. So you can see here some of the data on the T cell, for example. This is a B cell receptor. So we enrich for the B cell receptor, and it's again very specific to the B cell. And here we enrich for the T cell receptor, again very specific for almost all the T cell having the receptor expressed. Finally, I just want to talk about Dinoscope for a minute. So this is a way that you can use the data, and if you treat your cells or your sample or your mouse with this compound, then you can distinguish the RNA that was produced after the exposure to the compound. And what does that mean? So for example, in this case, if we focus on the B cell in the sample, you can see that a lot of B cells are having new transcript compared to, example, the fibroblasts, that most of the cells just have old transcript. So you can see like how active your cells are, but you can also focus at the gene level, where in green, again, for those B cell, you can see new transcripts on some genes, not all the genes, or old transcript. So those genes are basically were already expressed in this quantity before treatment. Those two last genes, they basically stop being expressed by the treatment. So it's very interesting to have this temporal resolution of your transcript so you can understand better what a certain treatment will do to your cell or your mouse. And we can study, we talk a lot about human cells and mouse cell, but we can study plants. So this is an example of what we did with the protoplasm of some of the plants. So we did Arabidopsis leaves, and we did Arabidopsis roots, and we did some Saccharomyces cerevisiae here, which is the budding yeast. So we just had to tweak a little bit the lysis to accommodate single cell for yeast. And we had a customer that was quite interested in doing so to study the yeast that they were doing for brewing beer. So that was one of the application in the food industry. But yeah, I don't want to go into more detail. Maybe I can stop here and take some questions. And this is just sightseeing experiments. Yeah.
So thank you very much, Julie, for the presentation. It was very insightful. And I'm sure the participants might have a lot more questions to ask, but in the time being, we have been asked by Stephen Willis in the chat about how many target probes can we have for focus scope for the mutations. And
Yeah. Okay.
yes, yeah, and I answered it in the chat, but it would be nice for everybody to also hear it from you.
Hopefully I have the same answer as you. Yeah, so I think it's about 10 different targets
Yes.
or 10 different genes.
If anybody has any more questions, please feel free to type or let us know in the chat. I think we have the Q&A section as well as the chat, so you can use any of it to type in your questions. So I think I cannot currently see any question right now popping in. So I was thinking probably you can write in the chat about whether you are planning for doing single cell, single nuclei, or any of the novel kits that Julie mentioned. I think she mentioned quite a lot of different multi-omics options. So one was about capturing the glycosylation proteins along with the transcriptome, then with the target sequences, which I think is pretty cool, and with the temporal resolution along with the transcriptome so that you can get like a time resolution across. So which one sound interesting to you? You can please feel free to write in the chat. Be happy to hear about your opinions.
Maybe people are hungry.
Yes. Okay. Yeah, then if there are no more questions, maybe we can wait a minute if anybody has, and then we can stop the meeting in like a minute around 6 o'clock. So we are almost on time. I think we have a question. Yeah, so Stephen is asking, do you think you will expand the amount of targets in the future?
For the focus scope. Yeah, it's a little bit of a tricky one because more you dilute if you start diluting too much your poly T, then you may start affecting the sensitivity of the transcriptome. So if the researcher were willing to do so, then we can put more targets there. There's no problem. I think you just have to find a good ratio to not affect the transcriptome. So it's a trade-off.
Stephen, you can let us know if that answered your question, and if you have any more questions regarding focus scope, you can also type it in the chat. Yeah, and I think one thing that you mentioned shortly in the end about site-seq was also quite nice. I think that even though we don't have it as a, you just need the single cell kit and the antibodies, and then you can do the site-seq experiments.
Mm-hmm. Yeah, so the current product is compatible with Total-seq A. So Total-seq A will have antibody that are conjugated with a barcode and a poly A, and then the beads have the poly T, so they will capture this oligonucleotide, and then it's quantifiable, so you can know which cell are expressing CD45 or things like that. So yeah, we tested both the BioLegend antibodies. They have a cocktail ready, or you can customize your pool of antibodies. So we tested that, and we also tested the AbSeq immunopanel. So I think it's like 30 different antibodies, and they really went there with white gloves and put the good ratio of an antibody, so there's not like one antibody that take over. And that worked really well too.
Sounds great, I think. Another question that we have right now is from Monica Toma. Is there a way, or do you think of ways to develop single cell protein analysis within the cell, not only the surface markers?
Yeah, there's a lot of publication how to permeabilize the cell so the antibodies can go in. But again, it's more publication. It's not a product. So it may not work with every cell type. Like everybody that published this kind of new technology often use PBMC. And then it will work for PBMC for almost everybody. But then you go into something more tricky, like a tissue, and then it becomes more difficult. And before it becomes like a sturdy product that we can sell, that people are successful with, then it's another story. You know, there's publication. People have been doing in site-seq. So it's doable. But yeah, again, it's not currently a product.
Sounds a bit challenging then.
It is challenging. Yeah, some people have been using nano antibody, like nanobodies, like the smaller antibodies that can go in, so you don't need as much space. But nobody made it a product yet that I know of.
No worries. I think it's still like good to know about the Insight-seq technology. We also have another question from an anonymous attendee about how much will the sensitivity of the results be affected if we populate more than 10% of the wells? Has it been tested?
Yeah, so I mean, if we want to populate, if you want to target more cell, we have this higher density chip that has 250,000 wells. So then the proportion of the well occupied by cells is better. And then you can go with capturing up to 30,000 cells without having too many doublets. However, yeah, if you start loading too many cells, the chance that you have 2 cells per wells are going to increase. And then it's not single cell anymore. So
yeah, the 10 to 15% works best from like the ratio works best with our technology right now.
Yeah. So what people have been doing to go around that is they can identify the wells that have 2 cells, and there's some software that can do that, and it basically throw out those data. Or you can tag it, like there's antibody tagging you can do, there's cell hashing you can do, or we have this like technology that again covalently bind tags. And then if you multiplex and then you have 2 cells per well, because they have 2 different tags, then you can tell. But again, you're sequencing them and you have to just reject the data. So it's not cost effective for me to do that. And it just kind of make it harder on the data analysis. So I will load 2 chips before I have to load more cell on a chip. Yeah.
Okay. Yeah, I think then we answered all the questions that we had right
Okay.
now. And we also kind of, I think, are at the end of this meeting. So I would like to request everybody who is here online to actually go through the couple of questions we have after the webinar. You can let us know your feedback. If you have any questions or if you have any feedback for us, please feel free to write it and we'll be happy to stay in touch. You can contact me. My email address is also the same as my name and at singleron.bio. And we can also send you emails afterwards based on my team will reach out to you. But just to, if you would like to get in touch before that, you can always send me emails. And thank you, Julie, for the talk once again. It was great having you.
Okay. Thanks for hosting, guys.
Bye.